Well, hello everyone. Welcome. I am Avalon Bristow. I'm the director of the Mid-Atlantic Regional Council on the Ocean, or MARCO. I want to extend a warm welcome to all of you for the second of four webinars in MARCO's Microplastic webinar series. Today's webinar focuses on how microplastics transfer through the marine food chain. It's an important topic in discovering where microplastics end up and in which animals they interact with. I would like to extend another warm thank you to the members of Marco's Marine Debris Work Group who put a lot of time and thought into creating this series, and some of them are here today. The work group is made up of state and federal agencies as well as non-governmental organization partners, all of whom are working together toward a shared goal of bringing together regional stakeholders to collectively identify, understand, prevent, and mitigate the impacts of marine debris. In this webinar, we're going to highlight work and research that's helping us understand how microplastics are ingested by and accumulate in animals that are important to the Mid-Atlantic region, like horseshoe crabs, shrimp, and even humpback whales. We'll then have a facilitated discussion and question and answer session where we encourage you to ask the expert presenters who are here today questions about their work and also explore how regional partnerships like Marco can help advance efforts to prevent and reduce microfibers and microplastics in the Mid-Atlantic. Before I pass it over to our moderator, I do want to give just a few quick notes on housekeeping. We are recording this webinar and it will be posted to the Marco website after it's completed. Please keep yourself muted. There will be an opportunity for question and answers following the facilitated discussion portion of the webinar. You can submit your questions in the chat box at the bottom tab of the Zoom window. This webinar is all about offering collaborative opportunities, so do feel free to put your name, organization, and where you're tuning in from in the chat so we can help you make some connections if appropriate. I am very pleased to introduce all of you to our moderator today, Donna Morrow, who will go over a few other housekeeping items before introducing today's speakers. Donna is the director of the Center for Marine and Coastal Stewardship within the Chesapeake and Coastal Service of Maryland's Department of Natural Resources. She has served on the Marco Marine Debris Work Group since 2017 and sits on the Chesapeake Bay Program's Plastic Pollution Action Team. She leads efforts to reduce and recycle plastic waste from the marine industry, with whom she works closely with in her role as coordinator of Maryland's Clean Marina Initiative. She has worked at the Department of Natural Resources for close to 27 years and remains passionate about addressing marine and coastal environmental issues. I myself am a very big fan of Donna Morrow and truly thank you for participating today as moderator. Donna, go ahead.
Well, thanks, Avalon. I appreciate that. And thanks to everyone else who is online attending today. We have three excellent speakers who are going to discuss the transfer of microplastics at different scales and in very different species. I'm excited to hear from all of them. I'm going to introduce our speakers now, and they will each have 10 minutes for their, about 10 minutes for their respective topics. And then we'll have a facilitated discussion followed by questions from the audience from the chat box. Joe will be helping monitor that chat box with me, but we'll get there. And I'm going to go ahead and begin. So first we have Dr. Mark Botton, Professor Emeritus of Biology in the Department of Natural Sciences at Fordham University, Lincoln Center in New York City. Dr. Botton's students have studied the effects of pollutants, including heavy metals and organic compounds, on embryonic and larval horseshoe crabs, and most recently have documented the occurrence and potential trophic transfer of microplastics from horseshoe crab eggs to shorebirds in Jamaica Bay, New York. Botton is the co-chair of the International Union for the Conservation of Nature's Horseshoe Crab Specialist Group, which advocates for the science-based conservation of horseshoe crabs and their essential spawning and nursery habitats. Next, I'm happy to introduce Mr. Bob Murphy, a marine ecologist who specializes in fish ecology and habitat assessment and restoration, with extensive experience in freshwater and coastal systems. Mr. Murphy initiated a workshop funded by the Chesapeake Bay Program to evaluate the state of the knowledge, gaps in data, and potential management solutions for microplastics in the Chesapeake Bay watershed in 2019. He was the lead author of the workshop report. He continues to serve as an expert in microplastic occurrence in the aquatic environment, including field studies evaluating abundances in fish, submerged aquatic vegetation and habitat, source tracking, and trophic transfer. And our third speaker is Dr. Shirel Kahane-Rapport, assistant professor at Old Dominion University, where she studies the ecophysiology, biomechanics, and fluid dynamics of large filter-feeding marine vertebrates, particularly baleen whales. Her research focuses on the mechanisms of filtration in baleen whales, recording baleen whale behavior through biologging and microplastic ingestion risk and trophic transfer in Chesapeake Bay ecosystems. So thank you all so much for being here. And I will go ahead and pass it off to Dr. Botton.
Thank you. It's a pleasure to speak with you this afternoon, and thanks for attending, and we'll get right into it. So this short talk is going to discuss some of the research that I've done with my students on the potential for trophic transfer of microplastics from horseshoe crabs to shorebirds via the ingestion of horseshoe crab eggs. No surprise, we've all seen pictures like this from around the world. Plastics are in all marine habitats, enter through a variety of sources, and generally speaking, what we refer to as microplastics are particles less than 5 millimeters in size. Sometimes you hear the term nanoplastics also. Those are in the range of 1 nanometer to 1 micrometer. And the field of microplastic research has really undergone exponential growth. This is the number of journal articles searched through Google Scholar using the search term marine microplastics. And what you can see is that the trend is basically following a typical exponential curve. The last 5-year period is only actually 4 years. So this will no doubt end up being higher. But this is a field that's become increasingly well studied in the marine environment. However, with respect to horseshoe crabs, there are relatively few previous studies. A study in China found microplastics occurring in digestive tracts of juvenile horseshoe crabs. There, a number of laboratory studies showing adverse effects from microplastics as well. But again, in terms of literature on the American horseshoe crab, nothing had been done until the work that we did with our group. Where do horseshoe crabs and shorebirds connect? Well, horseshoe crabs, as many of you, particularly in the mid-Atlantic, would know, horseshoe crabs spawn intertidally on sandy estuarine beaches, deposit the eggs, and then the reworking of the sediments by other horseshoe crabs or by wind and waves exhumes the eggs from the sediment where they become easily available for shorebirds feeding on the sediment surface. And this is an important food source for shorebirds in a number of locations, best studied in Delaware Bay, but also, as you'll see, in our location, which is quite different. This is an urban estuary called Jamaica Bay, located in New York City, spans both the boroughs of Brooklyn and Queens. Our primary study site is a location near the mouth of the bay called Plum Beach. So our basic questions were to first see how prevalent microplastics were in the horseshoe crab eggs. Are the microplastics incorporated into the eggs during egg formation or oogenesis, or possibly in addition as a result of contact with the beach sediments? And then how important are the horseshoe crab eggs in the diet of migratory shorebirds in Jamaica Bay? And does the consumption of horseshoe crab eggs pose a risk to the shorebirds in terms of accumulation of microplastics? So this part of the study was done by a student who's actually listening in to this webinar. And she collected unfertilized eggs, as you see in the left panel. You can kind of massage the gonopores, and a female who's ready to spawn will also often spurt out a number of eggs, and those are unfertilized. And we can also unearth clusters of eggs from the sand, which we know are fertilized eggs. Another student, Emily Casper, collected shorebird fecal pellets and did a number of analyses with that. And so she sat in a blind and watched the shorebirds feed. And then once they left, she could go out and carefully collect the fecal material from the areas where the birds were feeding. And what you can see is that there's a seasonal aspect to what the birds are feeding on, and it's correlated with the abundance of horseshoe crab eggs. In other words, early in the season, when there are very few horseshoe crab eggs, the fecal material contains other invertebrates, but not horseshoe crab eggs. And then as the eggs become more abundant, reaching several thousand eggs per square meter, the birds flip over and concentrate their feeding on the horseshoe crab eggs. To analyze for microplastics, we used a modification of the Nile red technique. Samples are first treated with oxidizing agent, in our case 30% hydrogen peroxide, to remove organic material. We treat them with Nile red, and then under the fluorescence microscope, take pictures, and the microplastic particles will glow a greenish color using image J. Then we can then quantify what we have, and that's how the samples for the eggs, as well as microplastics, as well as fecal pellets, were analyzed. So these are some examples of the kinds of photographs we obtained. The green glowing material are microplastics, and then on the bottom picture is an example of some microfibers, which were also abundant. If we look at the numbers of microplastics in the different types of samples, these are average particle counts per gram of material, and you see fairly high numbers in fertilized eggs, even higher in the unfertilized eggs, which is a bit of an anomaly. And then microplastics also present in shorebird fecal pellets, but in lower numbers than what were in the horseshoe crab eggs. So we can then do a little bit of back of the envelope calculations to estimate the trophic transfer between horseshoe crab eggs and the shorebirds. So we take as an approximation the number of microplastics per gram of a little over 600. That's basically just saying, okay, we'll take the average of the microplastic counts in the fertilized and unfertilized eggs. We know how many horseshoe crab eggs there are per gram from previous literature. It's a little over 100. And then you can estimate the number of microplastics per individual egg. So we take 633 microplastics per gram divided by 104 eggs per gram, a little more than 6 microplastic particles per individual egg. There's also literature on feeding rates, and these are studies done from the literature. Daily consumption of horseshoe crab eggs is extraordinarily high, or can be estimated between 8,300 and 18,000 per day. Now that sounds crazy, but if you ever watch these birds feeding, they're just taking peck after peck several times per second. They're just like machines out there. So even though these numbers seem extraordinarily high, they do make sense if you watch the shorebirds and their foraging activities. So we combine all this information. We say, okay, there's 600, it's a 6.25 microplastic particles per egg, and a bird, let's say, eats 8,300 eggs in a day. That projects out to nearly 52,000 microplastic particles ingested per bird per day. Now, don't get too crazy about this because there's a lot of built-in assumptions. For one thing, the ingestion rates of 8,300 plus per day are based on studies of shorebirds in Delaware Bay, which has a much higher density of horseshoe crab eggs than Jamaica Bay. So the ingestion rates in Jamaica Bay are probably a bit lower. We also don't know whether microplastics are found in horseshoe crab eggs from other locations. My guess is that they would be found, but we haven't actually studied that. And it's since birds are not feeding exclusively on horseshoe crab eggs, particularly early in the season, we'd like to know whether microplastics are found in some of the other invertebrates that these shorebirds are consuming. But if you recall from the bar graph, there are fewer microplastics in the fecal pellets of shorebirds compared with the microplastics in the egg. So that raises a number of other potential concerns. Are all the microplastics actually ingested, or do they regurgitate some fraction? And of the ones that are ingested, what fraction are assimilated? We don't really know that. And the bottom line question is, if some or potentially most of these microplastics are assimilated, what effects physiologically do they have on the shorebirds? We don't know that either. So to summarize, microplastics were found in all samples of horseshoe crab eggs and shorebird fecal pellets from Jamaica Bay. Microplastics evidently transfer from the body of the females to their eggs during egg formation. And spring migrant shorebirds in Jamaica Bay concentrate their foraging on horseshoe crab eggs and therefore have the potential to ingest a large quantity of microplastics. And I thank you for your attention, and particularly would like to thank two students who did this research. Emily Casper on the left did the shorebird fecal pellet collection and analysis of material, and Royal McMahon Moore to the right did all the microplastic analysis. And I'd also like to thank various agencies for the permissions to do the work and funding sources from Hudson River Foundation and Claire Booth Root Scholarship Fund. So thank you.
Wonderful, Mark. Thank you so much. That gave me a lot to think about there. Already starting to develop some questions. Next up, I'll introduce Bob Murphy once again, and he can give what he has for us today. Thank you.
Thanks, Donna, and thanks to Marco for the invitation to present this webinar. It's very exciting. Let me pull up my presentation here. And let's go to... Okay. I'm assuming everyone can see it, so I'll continue. I'm going to focus in on a lot of the work that's been done in the Chesapeake region, and in this case, focus largely on the Potomac River estuary as a microcosm of the rest of the Chesapeake Bay in the Mid-Atlantic coastal region. I'm not going to reiterate what Mark has already laid down as the basics about plastics, other than demonstrating that plastics, of course, are a global problem. This is a multi-data graph here showing the plastic production in the blue histogram columns increasing exponentially over time since the 60s, paired with the growth in population, the orange line that's also growing at the same rate, essentially. Maybe a little bit slower than the plastic production itself. And interestingly, this graph also shows the introduction of laws that were designed to regulate plastic really plastic pollution, not necessarily plastic production. And you can see those laws were first introduced in about 2008, 2009. I don't know if there's that little dip in plastic production was related, probably not. But you can see that the laws are still pretty minimal when it comes to regulating plastic pollution. So we're going to pivot to the Chesapeake Bay and the Chesapeake Bay program. Donna mentioned this earlier, the Plastic Pollution Action Team. This was formed at the conclusion and recommendation of a workshop held by the Scientific and Technical Advisory Committee of the Bay Program, just to assess the state of knowledge. What do we know? What do we don't know about microplastics in the Chesapeake region? There are the final report that summarized the output from the workshop had 4 recommendations, as noted here, and which came one of the things that came out was the formation of the action team itself, affectionately called the PPAT, but it's the Plastic Pollution Action Team. And right now we're going to focus on bullet number 2 here to support research to understand microplastic pathways in the Bay, which is the theme of this webinar, looking at trophic pathways and organismal uptake. In this case, we specified things such as striped bass, blue crabs, oysters, and other critters. So how do we get here? This is important because we tried to follow a very logical progression of information gathering. Some of it was desktop literature reviews, then transferred into field studies. So the State of the Science Workshop was held in 2019. We had some milestones, as noted here, with a risk assessment. The risk assessment focused on striped bass and young of year striped bass, and we specifically focused on young of year striped bass in the Potomac. So this is kind of how we narrowed down a very large idea to something that was a little bit more attainable in terms of data and just scale. So where we are here in the 2024 and 25 we're now looking at the source tracking of microplastics in the systems and how they enter the food webs. A study we conducted a few years back, my colleague Ryan Woodland at the University of Maryland Center for Environmental Studies. We looked at first, just to grasp rather, how fish were across trophic levels ingesting microplastic particles. So we restricted the study to the upper tidal freshwater portion of the Potomac River and Anacostia River around Washington, D.C., as you'll see in the map on the left there. So a very urbanized area. We knew that there was a fair abundance of microplastics already in the system. And in this case, we wanted to look across trophic levels. So from the lower level planktivores, you see in the middle histograms here, those are fungulants, those are mummichogs for those in the fish world. The invertivores would be the next ones up. These are your sunfish, your bluegills. Then we looked at the benthivores. This is catfish. In this case, the very invasive blue catfish, which is everywhere. And I will point out that the blue catfish, not only does it ingest microplastics, but it ingests macroplastics as well. They have an extremely wide gape, and we found some very interesting things in their stomachs that were beyond the size classification for mummichogs. And on the top level, and this is most of interest when it comes to human health, is the piscivores. So in this case, we use largemouth bass at our top trophic level species. On the on the right here, we see we graphed the microplastics count on the x-axis, and what you see on the y-axis are just 3 different indices that are a reflection of body condition of fish, and we see the same pattern regardless of the health index used, is that as microplastics counts go up, the index for body condition essentially declines, deteriorates. So there is a relationship showing and demonstrating that fish are in poorer physiological condition with a greater body burden of microplastics, and that was across trophic levels. In addition to the trophic analysis, just looking at various levels of feeding, we also want to take the results from the original risk assessment conceptual model that looked at young of year striped bass. So we wanted to at least look at proof of concept were microplastics being ingested by young of year striped bass, in this case in the Potomac. The Potomac River represents the second largest spawning grounds for striped bass along the East Coast, the upper Chesapeake Bay being number one. So it's very important. Those who do follow these issues, striped bass numbers have been in decline over the last several years. A lot of it's due to poor habitat, but there is this idea that there are other factors that are also lending to the decline in the population. In the striped bass that we did collect, we found similar to the other trophic level species, about 25% of all young of year striped bass had microplastics in them. You can see this micrograph in the lower middle part of this slide shows the fibers. Fibers dominate microplastics in the Potomac River by far. It's probably 75 to 80% of microplastic particles are indeed fibers. We can thank our polyester fleece jackets probably for the lion's share of those. So once we've demonstrated now that the young of year striped bass actually contain microplastics, now we're very interested in taking the risk assessment conceptual model, the fact that we were able to demonstrate microplastic ingestion, and now to further evaluate. And I will preface this by saying this is an incomplete study, so I'm going to show you some preliminary data, but talk about it and its importance to our understanding of what's happening in the systems. Again, I want to acknowledge Ryan Woodland from the University of Maryland for his collaboration on this. The diagram here shows the pathways, the primary prey items for young of year striped bass. This came a combination of literature review and some recent genetic work that came out of Matt Ogburn's lab at the Smithsonian Environmental Research Center, where the three dominant prey items for young of year striped bass were bay anchovy, not surprising, Mysid shrimp, and amphipods. All very common organisms found in coastal areas in the Mid-Atlantic region. Mysid shrimp are of particular interest because, as well studied as the Chesapeake Bay is, we don't actually know a lot about Mysid shrimp ecology. These are not Penaeid shrimp, the kind that you eat. These are much smaller and different taxonomically, although they are crustaceans. We decided to look at mysids as our vector because there's a suggestion. There's some literature that has shown uptake by mysid shrimp and trophic transfer. So I'm going to talk a little bit about how we're doing that, where we are, and what we hope to demonstrate. As I mentioned, recent paper 2021, Hasegawa and Nicoya did demonstrate in the laboratory that mysid shrimp will take up polyethylene beads. It's important to note that shrimp, like most crustaceans, they masticate everything into smaller and smaller pieces. So when Mark mentioned the existence of nanoplastics, this is one of the pathways that a microplastic becomes a nanoplastic. It's just through chewing, essentially. Mysid shrimp are very important ecologically because they link multiple trophic pathways. They are omnivorous consumers. They live on both the benthos and within the water column. They exhibit diurnal migrations from the benthos and into the water column. So they really are the primary point or pathway from the media, in this case, sediment or water column, into the food chain itself. And they have been demonstrated as a trophic transfer vector for microplastics to fish and other systems. For those of you, it's always nice to show, these are what mysid shrimp look like. If you're young of year striped bass, that's breakfast, lunch, and dinner right in front of you in the Petri dish. They are found in swarms. They make up a considerable amount of the biomass in our coastal systems, particularly in estuarine waters. They like the brackish water habitat very much. So our approach was to first get an understanding of the plastic and microplastic body burdens that are found in the ecosystem. In this case, we did paired sampling between two major tributaries of the Chesapeake Bay, the Potomac, and also the Patuxent River, which is essentially the next river northward on the western shore of the Chesapeake Bay. Mysids were sampled, counted, identified, so we had a good understanding of the species present and their relative abundance, and most importantly, digested for microplastics extraction. These are collected using epibenthic sled, and you can see some of the stomach contents in that slide below. Thankfully, I was not responsible for dissecting the stomachs out of these critters. That's having students help with that is very helpful. Our estimated loadings by river. Here's just an example of where the sampling locations are. These are chosen semi-randomly. The constraints were salinity and bathymetry. So we're looking at collecting 3 to 5 meters depth of water, and basically downriver from the estuarine turbidity maximum through a salinity of about 15 to 16 parts per thousand. And those conditions exist in both rivers. Now, interestingly, we found stark differences between the river systems. You see the Patuxent on the 2 graphs on the right are the microplastics per river. So these are weighted by all the organisms that were collected. But we also have the microplastics count per Mysid shrimp on the lower one there. And you can see the Potomac River concentrations of microplastics are much, much lower. And this seems to be a signal, and we're seeing it in another study we're doing, we're conducting, looking at source tracking of microplastics in the Potomac. This is a result probably of dilution. The Potomac is a much larger system, more water, more dilution. And we're seeing that both, for the river-wide analysis in the upper graph, and then by river, by individual Mysid shrimp. And you can also see it a little bit in the lower graph, the station on the x-axis is as you move downriver. When you blow it up a little bit greater, you do have a trend downward, which also suggests dilution. So you move downriver, just a greater volume of water. But we didn't explicitly test for that, but the data certainly suggests it. So now we have a pretty good idea of the loadings found in the environment in Mysid shrimp. This is where I'm getting to. Where are we now? We are now in the process of taking it to the laboratory. So where the question mark is where we are. We'll be dosing Mysid shrimp. Nice thing is Mysids are very laboratory friendly, frequently used for toxicology tests, very widely widely available. And then we're going to feed those to young of your striped bass, quantify the trophic transfer, and look at some physiological responses at the same time, similar to what we did with the condition factors. Looking at the all the other fish species in the previous study. So that's where we are right now. I'd love to come back and present the findings of this when we have them. It's a very interesting study, I feel, and it's really demonstrating that the pathways that we're seeing in our coastal areas. Definitely want to acknowledge quite a few people who have been instrumental in making this work move forward. And of course, Ryan Woodland out of the University of Maryland, Chesapeake Biological Lab, and Kelly Summers and Matt Robbins from EPA. Then, of course, my staff at Tetra Tech who have been so helpful in field collections and laboratory analysis. And with that, I thank you. If you want to reach me after this, there's my email, and I will wrap it up.
Thanks, Bob. It was very interesting. I know I speak for all of us when I say we really look forward to seeing the results of that work. I think that's going to be very interesting. Okay, next up is Shirel. Are you ready for us there?
Yes, thank you.
Great, thank you.
I will share my screen, hopefully successfully. Okay.
Yeah.
Sorry, there we go. A bit of a lag.
There you go.
Are we good? Cool. Thanks, Joe. I see the thumbs up. Thank you. Yeah, thank you for the introduction. I'm excited. Hopefully I can wrap this up and we can have lots of time for questions. I am Shirel Kahane-Rapport. I'm a new assistant professor at Old Dominion University in Norfolk. I'm super excited to be a part of this and get to meet all these cool folks. And I'm going to talk about microplastic ingestion risk here, there, and everywhere. And hopefully it'll become clear where I mean. I guess I was just going to throw in a little bit about myself. I've been studying whales for a very long time using lots of different tools, lab-based ones and field-based ones. And what's been pretty cool about working at ODU is I've slowly had a chance to combine those things into some of these projects. And so I'll begin here with filter feeders, which cover a massive range of body size, from blue whales to manta rays to oysters and little shrimps and even flamingos. And we all know a lot about microplastic and human pollution, but I really believe that filter feeders are uniquely susceptible to microplastic pollution, compared to lots of other organisms. There are lots of pollution problems, but I think microplastic is really the one that hits filter feeders the most. And I think that can happen in a lot of different ways, but primarily through developing biofilms on microplastics, through the transfer of chemicals from microplastic breakdown once those microplastics or microparticles are ingested, and through trophic transfer when something else eats, just like Bob was just talking about, when something small eats something else and how it affects their body condition and whether or not they're full and what kind of a prey they become once they've eaten microplastic. I think those are the primary ways to affect filter feeders that eat microplastic. And I, of course, am interested in the largest filter feeders that are out there, whales, and particularly the baleen whales, the lunge filter feeding baleen whales, the rorqual whales, they're sometimes called. And so there's this amazing map. This was generated by Germanov et al. It's in the journal Tree from 2018, if anybody wants to look it up. And what this map shows is the overlap between whales, so those are those hatch marks, and microplastics in surface waters. Now, this graph really got me and my collaborators thinking, but one of the things that it got us thinking about was actually the challenges that the things that this map might be missing. For example, the highest microplastic concentrations in the surface are at these lower latitudes where we know whales don't often feed. They more prefer high latitude productive cold waters. And this all reflects the surface. And from having done a bunch of work with whales previous, I know that some foraging occurs at the surface, but a lot more occurs at depth. So while this map led us on a start, we realized that it didn't really capture some of the overlap that we felt really mattered. And so in order to target the overlap, we first went out and tagged some whales. So this is a tagging obligatory whale video, of course, because they're so cool, I think, of us tagging a blue whale in Monterey Bay. Hopefully this video will play. This is a tagger putting a customized animal tracking solutions tag on the back of a blue whale in Monterey Bay. And just a great video of a blue whale in Southern California. This is from BBC's The Hunt. And I put this video here to really show the absolute enormous volume of water that these whales are ingesting and filtering. And so we start off by tagging these organisms and understanding more about how much water they filter. These are the tags that we use. These are customized animal tracking solutions tags. They're called CATS tags. They stick on with a suction cup and they last from anywhere between 6 to 18 hours. And they give us extremely high resolution data of everything that the whale is doing while wearing the tag, sort of like an Apple Watch, but for a whale. And then next to this, I'm showing how we took microplastic samples. So there was one great study, Anela Choi, in 2019, but many other studies studied microplastic in the water column in California. And so what we started to develop is this overlap between tagged whales in California and microplastic sampling in California. And this map is trying really to highlight that overlap. In the triangles, you see plastic sampling, those red triangles, both at depth and at the surface. And the tags are indicated in those circles, and different colors represent the different species that we looked at: blue, fin, and humpback whales. So we looked all up and down the coast of California, from San Francisco all the way to San Diego, trying to collect microplastics. So some of that work myself and an undergraduate student did, Lauren Kashiwabara, who's now a PhD student, still studying microplastics. We collected surface waters, we collected water at depth, and lots of different tagged whales to try to see if we could show a spatial overlap between microplastics and whales in their environment. So one of the things that we had to recognize was that these whales are filter feeders. And so they're encountering a lot of water, as exemplified by that video of the blue whale swallowing that mass of water. And so we considered that direct consumption. wherein water may have floating microplastic in it, and the whales may be able to engulf that water and retain the microplastic straight out of the water. Another thing we had to consider was indirect ingestion. So if their prey, a fish or a krill, contained microplastic and the whale then ingested that, we consider that an indirect ingestion. So all this is going into our risk assessment of these whales' microplastic ingestion risk, where they're eating in the water column, what's in the water column, and what's in their prey. And so finally, we could overlay that. We could look at the direct and indirect risk due to where these whales were foraging in the column. So what this graph shows here, hopefully you might be able to see my mouse, is a division between whales eating krill, whales eating fish, and where microplastic in the water column is. And right away, when we laid these graphs right next to each other, we knew that we had taken that next step past that first map of where whales and surface microplastics overlap, in particular because of this area here. At 200 meters depth, whales do a lot of feeding. The bluefin and humpback whales, they eat a lot down there, and there is a very high concentration of microplastics floating in that 200-meter depth. And so this was a great confirmation that we are finally taking a good step forward into targeting where that spatial overlap between whales and microplastics is occurring. Then we had to consider something else, which is that collecting a lot of fish and a lot of krill, as you just heard, is super time consuming, and you need lots and lots of undergrads and help to do it. So instead, we trolled through the literature to look for risk assessment levels in Pacific ecosystems that we could then model, that we could then simulate. And here I'll just show our medium scenario. And in our medium scenario, what we settled on was 1 in 3 fish in a school of fish that a whale will consume has one single piece of microplastic in it. Whereas a krill school in our medium scenario, a swarm of krill, 1 in 20 in that swarm will have a single piece of microplastic. And we estimated that baleen might be able to retain about 50% of the microplastic from the water column directly. So these are our indirect numbers, and this is our direct number. And here on the next slide, I'll show you the results of our medium risk scenario. So what we have here in the red box plots, we have the indirect consumption. So the consumption that occurs from eating prey, the trophic transfer consumption. And in the blue, we have the direct consumption. So immediately your eyes may be drawn to the difference between the red box plots and the blue box plots. There is far more microplastic ingestion risk as a result of trophic transfer than there is as a result of retaining it from the water column. And these rainbow plots, these rain cloud plots, combine that information together. In gray, we've plotted the humpbacks. In orange, we've plotted the fin whales. And in blue, the blue whales. And what our numbers show is that a blue whale is eating about 10 million pieces of plastic per day. A fin whale would be retaining about 6 million pieces of plastic per day. And a krill-feeding humpback whale is retaining about 4 million pieces of plastic per day. And a fish-feeding humpback whale is near to 1 million pieces of plastic per day. So lots and lots of plastic retained per day. And this is just our medium scenario for whales eating in the Pacific. And so we tried to make that into a weight, because pieces of plastic per day is very difficult to conceive of. Like, have you ever really thought about 10 million pieces of something tiny? It's probably hard to wrap our minds around. So we tried to convert it into a mass, which is in and of itself hard, because if we all know a lot about microplastics, we know that the weight of a piece depends on what the piece is made of, the density, the size, the morphology. But we're estimating if a piece of microplastic is between 0.2 to 4 milligrams per piece, then a blue whale at 10 million pieces per day can be consuming anywhere from 2.5 to about 44 kilograms of microplastic per day, which is definitely not a weight burden that we should be excited that they are adding to themselves. So these are our broad results for our Pacific study. And that was the there. Now I really am focusing on the here, which is the Chesapeake Bay. I just got here, and I'm really excited to build off the work of the folks who are already here. And some of my colleagues like Jeannie Shearer, they published work in 2024 that showed where the whales are foraging in the Chesapeake. And I hope to continue to collect all that same data I just described for the Chesapeake. And I'm also hoping to do that in Azerfontein, South Africa. I was just there in January. We started the preliminary work. And man, is there a lot of plastic in that ecosystem. It's a bay system, so similar to the Chesapeake in that sense, but an upwelling system, so more similar to what we found. I think my screen just blacked out, hopefully not for everybody else. Very similar to what we found in the Pacific. So I'm super excited to talk to anybody who wants to talk about these things. And of course, I have to thank all of my funders and the many, many colleagues who are working with me and will be working with me in the future on these projects. And hopefully, I would love to add some more faces to these lists. And I have to shout out Joe for inviting me to speak here. So thank you very much, Joe. And yeah, thank you so much.
Wow, thank you so much, Shirel. That is really exciting to hear what the work from all three of you, the work that's taking place on this similar but very different species and locations, trophic transfer of microplastics. I'm going to start off by asking if any of the speakers have any questions for any of the other speakers before I jump in.
I definitely do.
Go for it.
I was just really interested in seeing those trophic transfer numbers that you were just showing, Bob, because that was very, very cool to see. Also in the horseshoe crab eggs, like the bioaccumulation thing is something that we're finding very hard to show in the whales right now, mostly because they're so large and it's like just difficult to collect samples from them. So, and the digestions are complicated because the samples are so fatty. So I'd love to hear more from both of you, how you're doing these dissections and plastic breakdowns and these counts too.
Mark, I don't know if you want to go on that one or
Take it first, Bob.
Yeah, the extraction of microplastics, that's the last part of your question, is actually it's difficult, time consuming, and what we don't have data on right now because it's expensive is polymer analysis. There's sort of a bottleneck of resource because it takes some very specialized technology to look at, what exactly are we looking at? Polystyrene, polyesters, et cetera. And that's really good to know when it comes to policy development. And but we can't always have that. We can make some inferences with the particle shape and morphology once they are extracted and examined visually, and that's actually really helpful. I think. I'm sorry, Shirel. What's the other part of your question?
Oh man, I was just so excited. It was like an incoherent question. I was also just excited about the Mysid shrimp because gray whales no longer live on this coast, but on the west, which is, I've worked with them a lot and they love Mysids. And now and we know that the Mysid shrimp in Oregon, for example, are like super polluted and the gray whales are definitely. I just wonder if maybe the humpbacks, like who's eating the Mysid shrimp other than the striped bass? Do we know? Like, could it be whales?
A lot of,
I don't know.
Not limited, it's not limited to striped bass. Think about it. They're just little crunchy little popcorn, for a host of different species.
Yeah.
So, and they're readily available. They're numerous. So it's easy pickings.
Totally. Cool.
Yeah.
Mark, did you have anything you wanted to add?
Yeah, I guess getting to the point of getting better data about trophic transfer. In the case of the shorebird work, the number of these species are federally protected. So, and I'm not about to want to go through the hoops to do dissections on protected species. So that's why we were limited to what was in the fecal pellets because, nobody objects to going out to the beach to collect bird poop. But by doing the fecal pellet, we don't really know, and I tried to make that point, that we obviously have some in the fecal pellets, and we have an estimate of what the birds are ingesting. But what's in there, what transfers from the gut of the shorebird to the other tissue where it might potentially interfere with various physiological or biochemical activities. And And that's not easy to do in any species, but certainly not when you're dealing with species that have protection.
Yeah. Yeah. I mean, we've collected feces from whales. I mean, they tend to poop right at the surface. It's very convenient for us. But like exactly like you're saying, it's really hard to make equal between what's coming in and what's come out. We're trying. That's part of the next goal. So I'd love to, I mean, you all tried, right? At least I'm curious. Yeah. We can talk later.
Okay. Well, thanks you both, all of you for that. I'm going to jump to the chat. We have a few questions coming in. And the first one is not directed at anyone in particular, so anyone who wants to take it. Based on your experience tracking species and collecting data, what is the most favorable way to track organisms, especially smaller ones like the Mysid shrimp? I guess that goes to Bob.
I guess a little clarification. What is meant by tracking? Is it just to assess the body burdens of microplastics? Is that the thrust of the question for tracking?
Let's see, that came in from Angela, and I don't know if she's going to follow up on that. Tracking species and collecting data.
I guess I would
to track organisms. Yeah.
I would sample, I guess it comes down to sampling and just following the same protocols that we've used to sample organisms historically. So for fish, we generally will seine in the beachy areas, electrofish up in non-beachy areas, freshwater. I showed in that one, mysids are interesting critters because they sort of live right on the surface of the sediments. That's why they can feed on what's settling on the sediment surface. But then they'll migrate into the water column. So I showed a picture of that epibethic sled. I think mysids are going to prove to be one of these species that are sort of the canary in the coal mine for microplastics because of that interface, because they're such an important food resource for species that we all like to consume. There's been a little work done in the Delaware Bay just to stay on the species part of this question, looking at blue crabs. Blue crabs will also ingest them directly, microplastics that is. Again, they kind of also live at that exposure nexus, being right on the surface of the sediments is where they typically inhabit.
Thanks, Bob. I'm going to go ahead and jump down to our next question in the chat. And again, it's for everyone, I guess. Have you, let's see here, do you ever find macroplastics? Have you ever seen evidence? I guess this is more of a question for Shirel. Have you ever seen evidence of whales ingesting entire bottles or bottle caps? Anything macro level?
I have not in a baleen whale. I am also holding a mug about beaked whales. I don't know if this is coming out backwards to you all, but a beaked whale perhaps may because they suction feed. The filter feeders probably, well, to be fair, it is extremely hard to dissect the stomach of a massive baleen whale. So I myself, and I think very few others have actually ever managed to like open that up and see what exactly is inside. But based on what I have observed, they very quickly spit things out that are not what they wanted. I've seen them spit out birds like almost immediately. They will rather like sacrifice an entire mouthful than accidentally swallow a bird. Ooh. They can, yeah, it's very strange to see, but like they immediately know it's in their mouths. So maybe they can do this with a bottle. Their throats are also, their esophagus is actually much smaller than you would expect for such a large animal. So depending on the size of the macroplastic, it simply would not go down. A bottle cap, perhaps, but I've not ever seen the evidence, but I wouldn't say it's impossible.
I mentioned, actually, it's funny you mentioned that, Shirel. We did find in two of our big blue catfish, and these have a massive gape.
Right.
They're omnivorous. They eat almost everything, including plastic bottle caps. They're not big enough to swallow a whole bottle, but we found bottle caps.
Wow, that's nasty.
It was reminiscent of
the scene.
It's reminiscent of the scene in Jaws where they catch the shark and the license plate comes out and it's.
Totally.
Yeah.
Well, thank you. Let's see, a couple more here. Question for you, Dr. Botton. Could the ingestion of microplastic contaminated horseshoe crab eggs impact the health and foraging behavior of migratory shorebirds?
That's certainly possible, but we have not studied that. With a lot of literature on microplastics, it seems as though the toxic or sublethal effects are often due to other things that adsorb onto the microplastics rather than the plastic itself. But that's an open question, particularly for the shorebirds in Jamaica Bay or elsewhere where microplastics are part of the shorebirds' uh, dietary intake. I'll couple that with the second question by the same person who asked whether the microplastics could be affecting morphology or inducing mutations in horseshoe crabs. All I can say is that for many years we have brought horseshoe crab eggs back to the lab and raised them in water from Jamaica Bay and or in artificial seawater in some cases. And the percent of development is very high. We usually get, 90 to 95% successful development, no obvious signs of anomalies. Of course, with the life cycle of horseshoe crabs, to raise them to adulthood simply isn't practical because it takes 10 years. But we can raise them in the laboratory through routinely the first two molts, and they seem to be perfectly healthy. But again, horseshoe crabs are incredibly resilient animals to all sorts of environmental pollutants. So they might not be the best bioindicator for the effects of microplastic contamination in Jamaica Bay. There probably are many other species with higher sensitivities to pollution.
Yeah, that's a very good point. And I think that kind of circles back to the original, the first part of that question about the impacts to the migratory shorebirds, if there have been observations or any plans to study them. If you had noticed any fatalities, then could you do necropsies and things like that? I don't know if you've gone down that road of looking at the next level up with the birds themselves. All right. For everyone, I guess, do you think there's an effective way to prevent is a different matter, but to clean microplastics from these large bodies of water? Is there anything new on the horizon there? They're very difficult, of course, to remove. So wondering if anyone has anything on that.
Yeah, very difficult to remove, Donna, as well as one of the focal points now is through removal through stormwater. So kind of before it enters the actual surface waters and coastal waters is through various designs. One of the big ones is actually using green infrastructure now, bioswales and things like that seem to be fairly effective at capturing particles before they enter the waterways. Once they're in the waterways and the sediments and things,
Yeah,
we'll have to give that some
the
serious
cleaning's
thought.
out.
Yeah, that's exactly.
I was having a conversation about microplastics with some Japanese colleagues a while ago, actually, and we're talking about how microfibers are so prevalent and that washing polyester clothes and stuff, and they seem very surprised that we don't have some sort of filtration system in the washing machines that would collect that material before it enters the body of water, because apparently that's standard in washing machines in Japan. So you've really got to grab them before they get into the water. I think once they're in the water, the removal would I don't know if it'd be technologically impossible, but certainly
Yeah.
be not feasible at all.
And if I may, I'll just clarify on that. The washing machine filters are probably the best attempt to prevent the discharge of microfibers from our clothing, but microplastics can be a result of macroplastics sort of breaking up once they're in the environment, or they're nurdles themselves, or any number of other sources. So we would never, I don't want to give any false hope that, washing machine filters, should that come to become standard here in the States, still wouldn't fully alleviate the issue. Joe, how are we doing on time? Pardon?
No, I was just going to add to the microplastic thing, but it's all good.
Yeah, no problem. I don't want to cut anybody off. Joe, how are we on time?
We have about 8 more minutes before we need to wrap up.
Okay, good. Thanks. I wasn't sure if we were a stop at 2 or if we could entertain a few more. Yeah, as you can see in the chat, Mark, there's one more question there for you. Can microplastic only transfer from female horseshoe crab to their eggs? Do male individuals contribute to their offspring regarding microplastics?
The short answer is I don't know, but I would gather it's probably not likely, because when the sperm fertilizes the egg, it's a very minuscule amount of material that actually enters the egg in comparison to the massive amount of yolk and protein that the female puts in the egg. But again, it's a testable idea. We just haven't done that.
Okay, thank you. Once again, I guess it comes down to volume, when you guys have talked a little bit about that in other settings too. Is there anything else there in the chat that I have missed? I don't think so. Here was an interesting question that someone else provided. Do we observe similar trends in microplastic ingestion and trophic transfer in freshwater and estuarine ecosystems as we do in the marine environment? How might the dynamics of microplastic pollution differ between near-shore coastal habitats and the open ocean, and what implication does this have for targeted management strategies in these areas?
The first part, yes, freshwater systems, every bit as much as marine systems in terms of organisms. Therefore, you make the case for the trophic transfer. What was the second part? Was managing to prevent movement in the open ocean?
Well, yeah, the dynamics of the microplastic pollution differing between nearshore coastal habitats and then the open ocean, and how that would impact targeted management strategies. What implications that might have.
Yeah, it's like turning off a faucet. It's I mean, my estimation, like Mark indicated, we've got to be really creative and thoughtful about how to develop a means to remove. I think the feeling maybe, particularly, Donna, when you're talking about the open ocean, I think the horses left the barn, because a lot of have already sunk. They've been incorporated into sediments. There's, the million years from now, geologists will be looking back and they'll see the layer that represents, the Pleistocene era. I don't. It's stopping the flow currently is what should probably be the thrust of our efforts. Cleaning up after the fact can come, but I don't think the technology exists to do that.
Yeah. Shirel, did you have any thoughts? Since I know a lot of your research has been in the deep water oceans.
Yeah, that we are making a very big effort because of that to identify the polymers that we're finding, because we're trying to determine, can we show, is it coming from fishing line and from fishing boats and from boat paint, the things that we're finding, or are they, tire pieces and agricultural runoff associated things like from a hose or from a tarp? And it really depends. The more coastal you are, absolutely, there's like a greater aggregation of plastic in the water in general. But on the flip side, whales and manta rays and whale sharks are organisms that filter like almost 365 days a year, hundreds of thousands of gallons a day. And so they're aggregating and aggregating and aggregating. And so I doubt that it will be equal, but they're basically filtering, you know what, I can't think of the word right now other than aggregating. They're doing the aggregating that like the currents around the shorelines do just in themselves. And so we do definitely worry about it for sure. There's also layers of plastic, as that graph I showed has shown. That 200-meter depth was out in the middle of Monterey Bay, 60 kilometers away from shore. There are like the Pacific garbage patch, for example. Yes, it's at the surface, but most of it's in the middle of the water column. Like it's quite deep. So I think there are probably like hidden secret pockets of plastics in gyres that we don't know about. And a lot of it is getting into the sediment too, but things trickle really slowly down through the water column once they start reaching haloclines and thermoclines. You know, that slows down the mixing of tiny inorganic particles. It's absolutely going to slow down buoyant particles. So I don't know. I'm very in the middle on that issue, I guess.
Yeah. Well, we don't know what we don't know, right? But I think Bob's point was well taken, which is that you want to stopping things upstream early and often is by far and away the least difficult, least expensive option that we have. All right. Well, I know we're getting towards the end of our time, and I thought I'd just put one final question to the panel. What opportunities do you see for greater regional collaboration addressing microplastic ingestion and its transfer through the marine food web? And are there any specific initiatives or partnerships that could help, pool resources or data and efforts to tackle this issue more effectively in our region?
I'll take a first crack at that. And one thing that I have noticed reading through literature is that there's a very big challenge in front of us in that different labs use different methodologies for quantifying microplastics. Even definitions of what a microplastic is kind of all over the place. So in terms of regional cooperation, I think it would be helpful to at least come up with a document of some sort that outlines best practices. It's very difficult to compare one study to another if the units of measure are not the same.
I can pick up on that, Mark. I think that's an excellent point. And Donna, you as a member of the PPAT in the Chesapeake region, that's the whole idea of forming the team, was to be collaborative and sharing resources and information. And one of the items that actually came out of that group that we contributed to putting together for EPA and the Bay program at large was exactly that, a standardization of units of measure for microplastics. So that folks, measuring, you're looking at microplastics in whales down at the mouth of the bay are using the same nomenclature and size classifications as those looking at uptake and critters, up in the watershed. And that was done for the Chesapeake Bay, but certainly is applicable elsewhere for sure. So I know I can speak in the Chesapeake Bay region. It's been pretty nice seeing the collaboration amongst a lot of different researchers. Just the common thread being microplastics as part of it. So it's nice when data can be put together and we can start looking at bigger pictures, I guess.
Yeah.
I just not so subtly plugged a paper that I was part of, a contributor to, which is it's a global plastic ingestion bioindicators project. It's part of the SmartNet program, which is part of the UN Plastic Goals initiative. And it's a paper that we worked very hard to have representatives from the entire, like every country with an ocean. We very much are like aiming toward having someone from everywhere. And I think we tried very hard, and I think we did quite a good job. Particularly like the global South is underrepresented in all microplastic research, it seems. And so this was a place where we had like 12 different time zones on a Zoom at the same time, but the goal was to say, like, what is the standard in India, in Chile, in South Korea, in the United States, in Canada, in England? How can we set these standards? What are the animals that we should focus on as bioindicators? Because I was just so excited hearing about Bob's methods and Mark's methods. Like, I want to learn more about those methods because the methods are changing all the time and new people think about great ways. And there are some groups, this being one of them, that is trying to like standardize those things. But it's super hard. It's super hard to do. Yeah, but there are efforts being made, is all I will say.
And yeah, that is wonderful. And that is a nice place for where Marco sort of has brought these folks together and looks at this issue at a regional level. I would encourage folks to stay in touch and revisit, and feel free to participate in any of those groups or future chat or future talks. And in fact, to that point, I did want to post a link to posted, so for the rest of the year. But in terms of microplastics, yeah, it's been wonderful the work that has happened in the Chesapeake Bay region. And Shirel, please get in touch with Bob on that and keep that conversation going. And it's been fascinating hearing from each and every one of you. It's really, really very educational opportunity, and we're really lucky to have you. Thank you so much for taking the time to join us. I think we all know that there's a lot more research to be done, and we continue to learn from each other. But with that, I think we've covered a lot of ground. I'm going to let everybody call it an afternoon and move on. But thank you all so much for joining us. We really appreciate it.
Thank you so much for having us.
Thank you so much for organizing it.
Thanks,
Absolutely.
everyone. Thanks, Donna.
Yeah.
Thanks.